Okay, so yes, as you said, I'm going to present this work that is just about to be published, and it comes from my PhD thesis. So the key question we want to ask is whether there is a neural circuit that is specific for making novel inferential choices as compared to familiar choices, for example. And we also want to bridge the knowledge that we have that comes from the neuroscience of value-based decision-making in the monkey and in the human, because you probably are aware that people studying value-based decisions in monkeys have generally focused on the orbitofrontal cortex. So coming from Padua Schioppa and more recently other studies have found that neurons in OFC correlate with the values of the options that are presented and allow to make the comparisons. Lesion studies by Murray, for example, and other labs have found that disruption of this area will impair decisions. But when you take a human and you do an fMRI study, you generally find that economic choice activates a different area, which is often called VMPFC, or in any case, medial frontal cortex extended to ACC. So is this a difference of species, or is a difference of task or a technique used and so on? This is a bit the question that we wanted to ask. These two areas are sometimes confused as if it was a single one, but if you look at anatomical studies, they have different connectivity profiles, with the medial frontal cortex being connected with the rest of the medial part of the brain, including cingulate and medial temporal lobe, whereas the OFC has a different profile. Now, you could guess that a monkey study is not quite the same as a human one, because the animal received a long training, and therefore its learning is gradual. So it will sort of slowly acquire the different elements of the task and the associations. So maybe this is what OFC is about. Whereas a human is generally taught the instructions for the task just minutes before entering into the scanner. And so the learning is much faster, and the task is performed for the first time at the time of the experiment. So maybe this is what the VMPFC is about. In order to test this hypothesis, well, let me explain better what I mean by novel choices with this example. So if you need to go on holidays and you can choose between these two beautiful Italian cities, but you have never been to either one, you can choose between Siena, which is a medieval town, is in the sort of on a hill area, whereas Syracuse is more in the south, it's on the seaside. So you can sort of make up your mind about the value of each option, even if you don't know them, by putting together the different attributes. So you may know whether you like or not the seaside, you may know about the food and so on. And so you create a compound value, and then you make your choice. Of course, this is not the case in 2020 because of the coronavirus, so you shouldn't go on holidays probably in the next few weeks. But to summarize, yes, we ask whether monkeys can perform these novel choices, whether they are recruiting a different neural circuits, and later we'll also ask whether this circuit is encoded by a grid-like coding that has been extrapolated from what we know about navigation in physical space. So the task works like this. You have a first training set where the monkey for a long time is trained to make binary choices between options that associate different colors with different amounts of juice. Then on separate days, they learn to make binary choices again. And now the options are associated with different probability of rewards. But know that in every case, one attribute is changed and the other is fixed. And so these two training sets form this sort of gray area in the more general space of all possible combinations. And so you can understand that the monkeys will potentially know about all the other areas, in the sense that they know the meaning of the color and the meaning of the dots, but they have never been train on these white spaces, so they've never been reinforced by choosing this specific combination of color and dot. We have three different experiments. The first one, when the monkeys make a binary choice into an fMRI scanner, so we can see the neural activity. Then the monkeys will do a single sort of response task where there is no choice. And this we use for addressing the question of what are the neural representations that underlie these novel options. And finally, outside the scanner, we disrupt the behavior using ultrasound, and we see whether the effect on the brain makes a difference in the responses. So to answer the first question, the monkeys can actually make these binary choices. So here on the right, you can see a decision pattern of 4 different monkeys. And you can see that when the value of the 2 options is close to each other, they are indifferent, but when one option is much better than the other, they choose the best option. So this is the case for novel choices like here that are visually similar to a familiar choice, but this specific combination has never been performed in the training phase. Now, what happens at the neural level? First, we put all the choices together, familiar and novel, and we use this regressor that is the chosen value minus the unchosen value. So we see whether the neural activity is modulated trial by trial by the difference that of the 2 options, which is the amount of value that we believe should drive the comparison, of course, because the task is to compare the 2. And you can see that, quite unsurprisingly, orbitofrontal cortex, which is the area where we know the neurons code value, has an activity for the difference between the 2 options. However, now we do another contrast, which is value difference in novel trials minus value difference in familiar trials. So we see whether the 2 different types of trials activate differently the brain. And we see that they do so in medial frontal cortex. So in OFC, the effect is similar for familiar and novel, but in MFC, the effect is maximally different between novel and familiar choices. So we argue that this MFC circuit has been recruited specifically by these novel choices, where the monkeys know the components of the options available, but has not a very long training that has sort of strengthened the association by reinforcement. Okay, so then we are asked, what are the neural representations that underlie these novel options? First, we use a repetition suppression design. I go quickly on that, but the design means that when you show the same thing twice, the brain will respond less on the second time, so there is a suppression. And we see that when you present the same stimulus twice, you have an activation in orbitofrontal cortex again, which is sort of similar to what we got before, a bit more anterior. And so we assume that this area encodes the identity of the options that are presented, but not the value independently of their identity. Now, in order to ask how this value is computed, we took this hypothesis for the literature on grid codes. So if you remember, when a rat, for example, navigates into a space, you will have some place cells that will activate, will have a receptive field in just one location. But then they will have a grid cell, usually in entorhinal cortex, that have an hexagonal pattern of symmetry in the receptive field, which means that the same cell will activate in many locations, but not all over the place, with a very specific structure. And now we want to see if this spatial value space that we described can be analyzed in the same way. And we found that there is indeed a signature in the fMRI that, well, the data are a bit noisy because we have very few choices before the choice will not be novel anymore and will become familiar again. But when you look at novel choices, you have a peak of activation precisely in this medial anterior region of PFC. And we test that this effect is specific for a six-fold symmetry, so the pattern is hexagonal and not for other types of periodicities. And we also tested by estimating this grid in two separate subsets of each session, there is a consistency. So we now know that in MFC, there seems to be a grid code that spans this non-physical space that monkeys are exploring when they see one option at a time. So they essentially navigate into this space, even if it's an abstract space composed by the amount of reward and the probability of rewards. And then to conclude, we decided to disrupt this network by using transcranial ultrasound stimulation that can be done without invasive surgery, but is equivalent in some sense to TMS, but can reach more deep brain areas. And by doing this, we asked the question of how the monkeys is going to combine the two attributes. So you remember that you have magnitude and probability, and the correct way to do is to multiply magnitude and probability and to estimate the value of, to choose the option that has the highest magnitude time probability value. However, you can take an easier way, heuristic, and you just sum the magnitude and the probability. So this is what you do whether you are unable to integrate the two properly. And we compared how much they follow one strategy and the other, and we noticed that when you stimulate this more anterior and central part of the PFC, you have a specific decrease in the amount of optimal choices made by the monkeys. In this case, we have only 3 subjects after stimulation. So to sum up what we have seen, MFC disruption impairs ability to base choices on integrated value, and therefore they rely more on the 2 separate dimensions.
Alessandro, yeah, okay, great. If you can wrap up in a few sentences would be great, thanks.
So you have the first experiment when we show that they can do these novel choices, and MFC has a different effect for familiar novel choices, even when you keep the difficulty the same. The second experiment showed that when you show one option at a time and you move around the space, you activate a sort of grid code. And the third experiment shows that when you disrupt this network, then the ability to make optimal choices is indeed impaired. I need to thank all my lab, and in particular my supervisors, Matthew Rushworth and Mira Klam-Fluger, as well as the other co-authors of the paper, David, Leonard, and Jerome. Thanks.
Thank you very much, Alessandro. Again, on behalf of all the audience, great talk and very interesting data again. So while again, I wait for the questions to come in the Q&A panel. So I found this like homology between the coding scheme you find in MFC and this grid cell type of presentations, quite fascinating. Yeah. But to admit, I have to say it wasn't very clear. Maybe you had to a little bit go fast on that. It wasn't clear to me how did you interpret it as actually as grid presentation somehow. Could you go back to that slide if possible, maybe just to...
You asking how you do the analysis to test?
Yeah, I mean, how do you interpret that as a grid type of coding?
So maybe I can, do we have a, yeah. Well, I don't have an image for that, sorry. But anyway, so here the idea is that when you move into this 2-dimensional space, you occupy different locations, right? And the options are succeeding each other very fast. Now, there is supposed to be a symmetry in the effect. So the only problem is that you don't know what is the orientation of the grid. So we do what is called an F-test, where you have the sine and the cosine of the trajectory. So at every single trial, we estimate this angle alpha, which is how you go from the previous option to the next option. And if either the sine or the cosine of this angle multiplied by 6 modulates the neural activity, it means that the neural activity goes up and down, depending on whether your direction is aligned or misaligned to the grid. So if you imagine going this direction, then this, can you see the mouse? Then you go.
Yes, yeah.
Every time you move along the grid, you will have maximum activation. And every time you move sort of obliquous, then you cross the white space and we'll have lower activation. So when you try all possibilities, you will see this 6-fold symmetry that goes up and down 6 times within 360 degrees. That's the way we tested based on previous papers. I think the first one of which was by Christian Doller.
Great, thank you very much for the great answer. And I was just wondering if you plan, giving that, I mean, I think again you compared that, I mean, you started by comparing this behavior of novel choices to human. So one of the advantages that you have in Macaques is that you can also go a little bit more in details. And are you thinking of other techniques, like really looking at then seeing your neural recordings and see if the FC can also see this type of coding at the level of single neurons or population of neurons?
I think, I mean, I didn't perform electrophysiology in the past, but this is one of the avenues we're exploring. I think that would be particularly interesting to have single unit recordings in MFC. And there are few people who publish results there. Many people say that the activity is relatively small compared to what you can measure in OFC, but probably they didn't try our task, so it would be a good thing to do.
Great. Thank you very much, Alessandro, again. And again, for the interest of time, we have to move on. And